SwayamPrabha. Digital India. Educated India.
Hello and welcome to today's lecture of Introduction to Mechanobiology. In the last class, I discussed about atherosclerosis. And so in this disease, you have buildup of plaques which limit blood flow through the arteries and eventually might manifest itself as a heart attack when flow is completely stopped. Okay? And I showed you how if the flow profile can change from laminar to disturbed. Okay. As part of being disturbed, it can be oscillatory or turbulent. And described this paper which showed how YAPTAZ activation by disturbed flow led to endothelial cell proliferation and inflammation. Okay. So, as part of my reading assignment, you have this 2016 paper Okay. So, you have this paper as reading assignment, flow dependent YAPTAZ activities regulate endothelial phenotypes and atherosclerosis. Okay. So, today I will discuss about another disease, okay, muscular dystrophy. So, this disease is a group of disease that cause, as you have the term muscular dystrophy. So, you have progressive weakness and loss of muscle mass. Okay. So, there are various types of muscular dystrophy, among them the most common being Duchenne muscular dystrophy, okay, MD. So, this affects boys and starts between the ages of 3 to 5. You have Becker muscular dystrophy. Okay. It is much milder than Duchenne muscular dystrophy. It also affects boys, but in the age group 11 to 25. Okay. So, its effects start to manifest slightly later. You have congenital muscular dystrophy. Okay. As the term suggests, it starts at birth or shortly afterwards. You have limb girdle muscular dystrophy. Okay. Typically starts in a person's teens, roughly age 20s. Okay. So, there are various other muscular dystrophies, including Emery-Dreyfus muscular dystrophy. Okay. Typically again affects boys around 10, okay, and these lead to heart problems. So, I will discuss about two of them, Duchenne muscular dystrophy and limb girdle muscular dystrophy. Okay. So, in Duchenne, okay, so if I draw, okay, so if I draw the muscle structure in most cells, okay, so imagine if this is a plasma membrane, okay, so on the outside you have, okay, okay, and you have this bigger complex, okay, which are linked. Okay. From here, okay, so in normal muscle, you have two sets of adhesions which link the actin cytoskeleton to the outside. Okay. You have the integrin based focal adhesions. Okay. If the integrin based focal adhesions, and you have the what is called the dystroglycan complex. Okay. So, in DMD or Duchenne muscular dystrophy, so as part of the dystroglycan complex, there are multiple proteins, okay, like gamma sarcoglycan, I am just writing, okay, sarcoglycan, as well as dystrophin. Okay. In DMD, due to mutation, dystrophin expression is completely gone. So, the dystrophin complex is missing. And in limb girdle muscular dystrophy, gamma sarcoglycan is missing. Okay. So, what has been observed? Okay. So, to study the effects of these proteins, what people have done is they have created mouse models where they have deleted the expression of either dystrophin, which is called an MDX mouse, okay, where the dystrophin protein is missing, or you have gamma sarcoglycan deficient mice. Okay. So, what has been observed is in gamma sarcoglycan deficient cells, these cells compared to normal wild type, these cells exhibit enhanced apoptosis. Okay. And this is nearly 10-20 times, 10-20 fold increase in apoptosis. Okay. To study the effect, what underlines this mechanism, what authors have done is they have created these patterns. Okay. You have, so in 2D, you will have these strips of adhesion islands. Okay. You have, these are ECM coated. Okay. So, if you zoom in, you will get these islands which are roughly 20 microns in width, okay, and 100 to 1000 microns in length. Okay. And the remaining area outside, okay, this is non-adhesive. Okay. So, later in lectures I will show you how you can make use of microfabrication to generate these structures. Okay. So, this allows you, so when you plate cells, the cells will go and populate these patterns. Okay. So, you have myoblasts which are aligned and they populate these patterns, okay, and these myoblasts which finally fuse to form multinucleated myotubes. Okay. You can generate these multiple structures. Okay. So, what people have done is then on glass you have these substrates where you create this bottom myotube, and on top of which you add a second layer of muscle cells. So, you get the top myotube. Okay. So, in essence you have created a cell on cell system. and so this is a top layer of muscle cells or which finally fuse to form myotubes, and this is the bottom layer. Okay. So, I had previously discussed the effect of stiffness on muscle cell differentiation. Okay. So, these guys they differentiate optimally on 12 kPa substrates. Okay. So, because these cells are cultured, the bottom myotubes are on glass, cells never striate. Okay. But the top cells striate. Okay. So, when these striation was compared, okay, in wild type, okay, in C57 wild type mice, cells isolated from C57 wild type mice and cells isolated from gamma sarcoglycan deficient mice. So, what was observed, okay, the top layer, okay, if this was the level of striation, these cells, okay, this is only the percentage striated of top layer. Okay. So, these cells exhibited lot more striations. Okay. So, striation is linked to contractility, right. So, since formation of striations is indicative of a formation of sarcomeres, functional sarcomeres, okay. So, this was assessed functionally using a relaxation experiment. Okay. The idea is very simple. Okay. You have a, let us say you have a long myotube on a substrate. Okay. These are your nuclei, and imagine, okay, you come down with a pipette and detach one end of the myotube. Okay. So, what will drive is this. So, when you detach, the force balance is perturbed, as a consequence of which this myotube will relax and finally come to some other state. Okay. So, if this was the initial length, this is some other final length, okay, and you can track this length as a function of time. So, L as a function of time t. Okay. So, if you plot L by L initial, you will start from a value of 1 and then you will get a curve like this. Okay. So, what they observed was for C57 cells, which is wild type, whatever be the relaxation curve, for gamma sarcoglycan deficient cells, the curve was somewhat like this. Okay. So, this suggested, so since this final saturation point, okay, the saturation point, so what you can do is you can fix these curves, okay, you can fit these curves using an expression, let us say y equal to 1 minus c into 1 minus e to the power minus t by tau. Okay. So, let us say you use so these are your actual experimental points. You use this, you fit it with the following expression, you can backtrack two values, one is c and one is tau. Okay. So, tau is a characteristic time constant of relaxation. And c is an estimate of contractility. Okay. So, as t tends to infinity, let us say, let us see this expression, as t tends to infinity, this value will give you a value of 0. So, 1 minus C is the value. Okay. So, in essence C is nothing but the final ratio to the initial ratio. Okay. C is, if you take the cell, the L final by L initial is what is C. Okay. So, it gives an estimate of how much it contracts. So, if the contractility is less, then L final will be comparable to L initial and you will get a lower value of C. So, in this case, so compared to C57, the gamma sarcoglycan deficient cells are more contractile. Okay. So, what you can conclude from this experiment is cells are more contractile. Okay. And if you look at these cells in culture, so what you would see is many of these cells. So, this is actually the pattern. Imagine that this is the outline of the pattern on which the cell was plated. Okay. So, many of the cells exhibit a profile like this, okay, where the center, if you compare the central width to the end width, okay, you would have, compared to control cells, okay, if you had 10% of cells, okay, whatever be the percent of cells exhibiting this phenotype, okay, in case of C57 cells, in gamma sarcoglycan deficient cells it is significantly higher. Now, what might lead to this effect? One is the contractility, in which the cell is pulling on itself, the cell is executing this increased amount of forces, as a consequence of which it is detaching under the effect of its own force. Okay. So, in terms of the signaling pathways which were activated, what was observed was the ERK-McManus pathway, okay, was upregulated in gamma sarcoglycan deficient cells. Okay. So, this is the status in gamma sarcoglycan deficient cells. One more thing which was common, so this kind of phenotype, so increased apoptosis, was not seen in MDX mice. Okay. MDX mice or muscular dystrophy mice, they do not exhibit this increased apoptosis, but there is a commonality between MDX and gamma sarcoglycan deficient cells across both of these. So, compared to control, integrin signaling is upregulated. Okay. And when I say integrin signaling, as part of this, so integrin beta 1, sorry, alpha 7 was increased in both. Okay. Paxillin, which is one of the integrin signaling molecules, was also increased in both, but it was increased to a much greater extent in MDX mice. And so, Paxillin has two phosphorylation sites at tyrosine 118 and tyrosine 31. These are increased in MDX mice. Okay. So, phosphoscreen revealed that increased amount of Paxillin signaling in MDX mice. So, what is Paxillin doing in these cells? So, what was observed was in MDX cells, okay, in cells isolated from MDX mice, Paxillin localized at focal adhesion was phosphorylated. So, you had both Y31 and Y118 phosphorylated, and more importantly its localization was sensitive to contractility. So, when you treat it with bleb, which is the inhibitor of myosin 2, then you have immediate delocalized. So, Paxillin delocalizes from focal adhesions. Okay. And if you remove, then again they come back to the focal adhesions. Okay. So, this amount of turnover of focal adhesions can be assessed using FRAP experiments or fluorescence recovery after photo bleaching. So, what you do is you take a cell which is transfected with, let us say, GFP paxillin. Okay. Let us say this is an outline of a cell. Let us say this is outline of a cell, and you have GFP paxillin adhesions. Okay. You focus on one small adhesion, okay, and you shine light so that it is locally bleached, and then you track what is the intensity at this point as a function of time. Okay. So, if you plot the intensity, you will have a curve something like this. Okay. So, you can backtrack what is the percentage recovery. So, in this case, for example, if you had a protein which was fully mobile, okay, then after you bleach, it will come back to the same value. Okay. But at focal adhesions, okay, this was observed in the cytoplasm where the protein was freely diffusing. Okay. In focal adhesions, its recovery is much less. Okay. So, if you track the time to recovery and the recovery time, okay. So, what they observed was compared to focal adhesions, the recovery time. So, this recovery time was much higher at focal adhesions compared to cytosol, and when you treated with blebistatin, the recovery time decreased. So, you had a net decrease in blebistatin, and this is in cytosol, suggesting that paxillin is interacting with other proteins at focal adhesions. Now, what was also observed was when paxillin was overexpressed, okay, there was an increase in the percentage of striation in vitro. Okay. So, you use the cell and cell geometry to track how many myotubes have striated. Okay. So, there is in MDX mice, okay, there is an increase in paxillin. Okay. Paxillin localization was at a phase was sensitive to contractility. Okay. And when you took control, okay, or wild type, if you overexpress paxillin, so this led to increased myofibrillogenesis. Okay. So, this suggests, okay, these experiments suggest a link between paxillin and contractility or hypertension. So, you can assess it functionally using experiments like the relaxation experiments that I suggested. Okay. Okay. And what was observed, if this was the curve for control cells, okay, when you overexpress paxillin, so paxillin overexpression, so this is L by L init, this is time. Okay. They relaxed by a lot amount. Okay. So, you can track, and when they treated with blebistatin, right, blebistatin inhibits contractility. When they treated with blebistatin, this was negligible because blebistatin relaxes all contractility. Okay. So, you can track this difference compared to brevistatin, how much is the increase in this length. Okay. And what was observed was Paxillin led to increased relaxation. So, Paxillin transfected cells were hypercontractile. Okay. And this can also be indirectly estimated using atomic force microscopy. So, later in the course you will have a detailed discussion of how we can use AFM for probing mechanical properties of cells. Okay. And what was observed was, so if you tracked the stiffness, okay, if cells, if this is the distribution you get for control cells, okay, when you overexpress Paxillin, the curve shifted to the right, means cells became stiffer. And for bleb treated cells, so this is with bleb, this is control, and this is with Paxillin overexpressed cells. Okay. So, this suggests that Paxillin transfected cells are hypercontractile and stiffer. So, the same experiment was done between, so all these experiments were done with C57 cells transfected with Paxillin. Okay. When the same experiment was done with C57 and MDX, okay, what was observed was compared to the response of C57 cells, okay. So, with in MDX cells they were more contractile, okay, but they were less contractile than gamma sarcoglycan deficient cells. Okay. So, you have a situation in terms of contractility C57 less than MDX less than gamma sarcoglycan deficient cells. Now, these guys are apoptotic, these guys are not apoptotic. Okay. What was also observed was compared to C57 cells, okay, tissue from C57, where the average stiffness was order 12 kPa, similar to normal muscle. MDX muscle tissue was nearly 1.5 fold stiffer at 18 kPa. Okay. So, like previously, MDX, which exhibit Paxillin overexpression, are contractile, are more contractile and stiffer compared to C57. So, what is the implication of this? Now, what has been observed is in the clinic, okay, MDX patients are often given this drug called prednisone. Okay. Prednisone. Okay. This is a glucocorticoid. Okay. So, this is used only as a palliative, okay, but how it works is not known. Okay. So, experiment was performed in which you had control cells which were treated with PDN or prednisolone, okay, and asked how does the stiffness change. Okay. What was observed was PDN treatment made cells softer. So, suggesting what has been done is using this drug you are reducing the level of tension. Okay. So, in essence what you saw is your MDX or gamma sarcoglycan deficient, in both these cases your dystroglycan complex is being perturbed, which leads to a contractile increase in contractility of these cells. In one case it leads to apoptosis, okay, in gamma sarcoglycan deficient cells, and in both the cases the tissue is stiffer. So, in both these cases this is associated with increased integrin signaling. But the problem is this integrin signaling is in turn driving hypertension, okay, promoting contractility and driver hypertension. Okay. So, this is a problem, though the integrins try to compensate the effect of dystroglycan by overexpressing integrins and integrin signaling, in essence it leads to hypertension. Okay. So, with that I stop here for today. As part of reading assignment, okay, I give you two papers. One is Griffin et al. Okay, Journal of Cell Science 2005. The other one is Sen et al. European Journal of Cell Biology 2011. Okay. Thank you for your attention.